Today we're going to start one of two lectures that we're going to have on genetics. Today we're going to talk about the molecular basis for genetics, that is, the things that happen on a cellular level that lead to the reproduction of the species. And then next week we're going to talk about Mendelian genetics, which is named after Gregor Mendel, which is the more macroscopic or life-size version of genetics, where we look at what actually happens to animals and individuals. I strongly recommend that you go over this lecture at least 2 or 3 times before we get together next week, because it has some fairly complex things that you have to take in, and so I would spend some time making sure you understand the various terms and the various processes that occur. So first of all, let's take a look at a definition of genetics. Genetics is the science that studies how characteristics are passed from a parent to an offspring. And so we will be talking about genes and how genes are passed on and how those genes happen to carry the various characteristics that we see, whether it's blue eyes versus brown eyes, or brown hair versus blonde hair, things of this nature. A genetic tendency is a term that's used for the range of characteristics that is set by our DNA. Now, genetics by itself does not set who we are or what we're going to be like or how we look in its complete way, but it gives us a certain tendency in terms of our height and various physical characteristics, as well as many effects on our behavioral characteristics. But there are other influences that impact the end result. The effect of the genes on who we are and what we are is called the genetic factors. These are the guidelines or the traits that are passed on through our DNA. Now, if we look at genes and chromosomes, first of all, we have to understand that not only do genetic factors take or have an influence, but there are also environmental factors. Environmental factors are those factors that are in a person's surroundings, that are not part of their genes, not part of the internal aspect of the person, but they're in the surroundings. So they're who their parents are, who their siblings are, their friends, their other relationships, their geography. And then there are also spiritual factors, and these are the factors in a person's life that are determined by the quality of their relationship with God. Now, the important part about this is that this many times impacts our choices, and our choices have a great deal of influence on who we end up being. So as we look at the genetic material, we have to define what a gene is. A gene is a section of DNA that codes for the production of a single protein, or sometimes more than one protein, or sometimes part of a protein. And this protein in some way influences a trait that we have. This protein may produce a certain pigment that influences our hair color, or a certain pigment that influences our eye color, or a certain process that influences how long our bones grow, things of this nature. And so an individual gene generally controls a protein or part of a protein, and generally controls, in many cases, one particular trait. Now, a chromosome is a network of DNA and its supporting proteins, and a chromosome resides in the nucleus, and we'll look at chromosomes a little bit more in the upcoming slides. But you can see here that you have the genetic material, and the genetic material is strung onto a histone, which is a protein, and then these histones are lined up and wound together and form a chromosome. So protein synthesis can be divided into two parts. Remember, I said that a gene is responsible for producing a protein. So we have to see the relationship between the gene and the protein that's produced. And so let's take a look at that in a little bit of detail. The first part of the production of a protein is called transcription. And transcription is essentially the making of a RNA, a piece of RNA that is built from the DNA. And the RNA is a mirror image of the DNA that it's built from. And so it's a negative copy, if you will. It's as if you look into a mirror and then you take a picture of the image in the mirror as opposed to your actual image. So let's take a look at this diagram and see how this happens. Here you have the DNA, and remember it's two strands and they're complementary strands, and the complementary aspects are determined by these base pairs, as we talked about last week in our lecture on that. And what happens is when it's time to make a protein, an enzyme comes in and unwinds the DNA, okay? And then another enzyme comes in called RNA polymerase, and it begins to add nucleotides like right here, and it uses this part of the DNA as a copy or a template to make this piece of RNA. And each of these are matched according to which base the template has. It's matched with its corresponding opposite. So for example, if this is an A, then this will be a T. If this is a C, then this will be a G, etc., which is why it's the mirror image or the negative copy of the DNA that's been unwound. And then as this is being produced, it's stranded out in this direction. So you end up with a long strand of RNA that is the negative copy of this strand, this complementary strand of DNA. So once it's built, it's then rewound, and then you have the DNA back the way it was. And so this whole process of making this RNA is called transcription. Now the second part is called translation. And translation happens when you take this RNA that's been produced, and this RNA that's been produced is called messenger RNA, and that's shown right here in this diagram up here. And don't worry about this right now, but there's a 5' end and a 3' end. Now this has come out of the nucleus, and the complementary copy of DNA is not in the picture here. And then a ribosome, which you see this says ribosome here, is kind of cut off, but it says ribosome there. A ribosome comes on and attaches itself in two parts onto this messenger RNA, and it begins to move from the 3' end to the 5' end. I'm sorry, from the 5' end to the 3' end. It moves in this direction, as it shows this arrow right here. And as it does that, then these transfer RNAs, which are shown right here, which are these big old molecules that have a special sequence of bases called anticodons, and they line up with the corresponding 3 bases on the messenger RNA. So the anticodon is a sequence of 3 bases on the transfer RNA, and the transfer RNA, let's move over to this one, it's carrying a amino acid. So it comes along and it has UUU, which RNA has U instead of T, and it has to bind to 3 A's. So it comes up and binds to these 3 A's right here, and then you have lysine that's attached to it. Then the second one comes along and it attaches, And this G has to have a C, this U has to have an A, and this G has to have a C. And so it attaches and it brings valine. And then these two guys attach to each other and they become part of the growing protein chain. Now these three bases on the transfer RNA are called anticodons, and the three bases on the message that the anticodon binds to are called the codon. So this sequence is called the codon, this sequence is called the anticodon, and they match up and bring the amino acid, and then this begins to grow, and this is called translation. And so the production of the messenger RNA, transcription, plus the production of the protein using transfer RNA, which is translation, those two processes together make up what's known as protein synthesis. And so here's a review slide. The part 2 was called translation. Now we didn't talk about this. We'll talk about this probably in another section. But the DNA first unzips and forms a messenger RNA, and this is called transcription. Then the RNA is used to produce a protein in what's called translation. So it's transcription and then translation. Translation is the process where a specific protein is used based on the sequence of RNA, and as you know, the RNA was made based on the sequence of the DNA. Now, the protein, actually the RNA that's produced is actually composed of two regions. One region is called an intron and one region is called an exon. Now the exons are the actual functional parts, or the part of the RNA that actually codes for protein. And the intron are the sequences of RNA that are between exons that code for nothing, and they act as spaces between each RNA sequence that codes for a protein. And so you have this intron acting as a space between exons, and the exons produce the protein. Now, one thing that I'll say is this direction of production to go from RNA by transcription to produce DNA by translation to produce protein was called in the early 60s the central dogma of biology. And it was believed at that time that all protein synthesis and all cellular processes involving DNA, RNA always went in this direction. Always went from RNA to DNA, from DNA to RNA to protein. And that was known as the central dogma of biology. Well, as it turns out, the central dogma of biology was not correct, because there were certain viruses that were discovered that only contained RNA and actually had something called reverse transcriptase, which actually uses the RNA template of a virus to produce DNA in the cell that it infects, and then that DNA is used to produce other RNA and protein and make new virus. And so this reverse transcriptase and this reversal made the central dogma of biology no longer central or dogmatic. And once this was discovered, it was found that indeed it could go in either direction. It can go from DNA through transcription to RNA through translation to protein, or with certain viruses it can go from RNA by reverse transcription to DNA and then back to RNA and then producing protein. So we'll stop at this point. This will be the first part of the biology lecture, and then the next part will continue with this discussion.